Hello and welcome to everyone joining us for today's Select Science webinar entitled Automation Enabled Assay Development for High-Throughput Screening. My name is Carrie Haslam and I will be moderating today's presentation. Today I'm delighted to be joined by Dr. Nina Grossmann, Lab Head at Merck, as well as Adrian, Schoen, Lab Technician at Merck. After the presentation, we'll move on to our question and answer session. Please feel free to ask any questions for the Q&A any time during the webinar, and you can submit your questions to the left of the screen. Also, throughout the webinar, we will be releasing some poll questions, so please do answer these as the webinar goes along. That would be greatly appreciated. So without further delay, I'd like to hand over to Nina for today's presentation, and I would like to thank her again for speaking with us today.
Yeah, thank you, Carrie, for this kind introduction. Hello, everyone. I'm sitting here in Darmstadt, Germany, together with Adrian, and we will give you some insights how we are developing assays for high-throughput screening, and we want to give a more introduction into how we are using benchtop automation for assay development. In this case here, we will introduce how we use the dragonfly discovery from SPT Lab Tech. When we talk about high throughput screening, I would like to introduce to you a little bit how discovery pharmacology at Merck in Darmstadt is organized. We have three departments here. One department is molecular pharmacology, the other one is cellular pharmacology, and we have the bioassay automation department. Molecular pharmacology is basically setting up biochemical assays, and cellular pharmacology is dealing with cell-based assays. And we are setting up these assays then to run in high throughput screening. And the bioassay automation group is then actually running these screens on the machines. So in molecular pharmacology, it's the biochemistry side of assays. We are setting up all kinds of different assays, such as protein-protein interaction assays, or functional enzymatic assays, such as kinase assays or enzyme assays for DNA or RNA modifications. In cellular pharmacology, assays such as imaging-based assays or reporter gene assays or co-culture assays, just to name a few, are being developed. To run a high throughput screening, of course, molecular pharmacology and also cellular pharmacology, we have to work hand in hand with our bioassay automation group to make sure that all these assays are in line with our quality standards. And these are depicted here. So in all of our assays, we want to have a signal to background ratio more than 3. We want to have a Z prime on the plate more than 0.5. The plate drifts should be lower than -20%. And what is really important for us, because we run these screenings takes a long time, the stability of the assay should be more than 24 hours. So when we run a screening campaign, we usually screen roughly 100 million compounds. In some cases, we run more than 1 million compounds when we include libraries from collaboration partners or libraries from other consortia. So we can end up to screen 1.5 million compounds in total. In the primary assay, the compounds are being tested usually at 10 micromolar single point concentration, and normally we do this on 1536 well plates. The hits from the primary screening, we will determine a certain cutoff, and these hits are then being screened in dose responses. Dose responses, that means that we test the compounds that we found in the primary assay, in the primary screening. We test these compounds usually in 10 different concentrations. And we run these in multiple assays in the secondary screening, of course, on our primary target. Usually we are very interested super early on how the mode of action of a certain compound is. This is why we include very early on in our screening campaigns some kind of mode of action studies. It could be a competition assay or a jump dilution assay. Then it can happen that in our secondary screening we include, when we run enzymatic assays, a certain domain of the target that we screen in the secondary screening to figure very early on out if we have allosteric binders or where the binding site of a certain compound is located. Usually we want to be very selective with our compounds. This is why we include in our secondary screenings already selectivity assays. And so we run this on a different target. And we have to be sure that we are not pulling out any technical interferers here. So what I want to mention here, we can run one screening, but in the end we will have multiple DRC runs, usually 3 to 4 runs. And this means for every DRC run, we are having a special designed assay for this. And often the clock is ticking, so we have to be really fast here in assay development. And I have mentioned on the slide before that we have very high quality standards. So we have to be fast, and we want to have a high quality in these assays. And as you see on this slide, we have multiple assays. So how can we speed up assay development for this multiple assays here? And we thought an easy way is to include very new equipment here. And one possibility to speed up assay development to use this new generation of benchtop dispensers. We already have dispensers in our labs. We have the echo acoustic dispensers in our assay development labs. And this is the working horse for us for compound dispensing. However, so until 2 years before, we didn't have dispensers that we could use for assay development. And then we invested into a new generation of these benchtop dispensers, and one of this was the dragonfly discovery. What was really important to us, that this dispenser has certain prerequisites, and we decided to acquire the dragonfly discovery for these reasons here. So the dragonfly discovery has a very high volume range. It can dispense all kinds of different liquid classes. We can dispense multiple liquids at a time, so we can do titration of multiple reagents in one experiment. We are really flexible here with the plates, so we can use it for 96 valve plates, but also we can do it for 1536 valve plates. If you have reagents that are very costly or where you don't have too much of the reagents, the dragonfly is really nice to use because it has a low dead volume. It has an easy software, so you can design your experiment on your computer and then just go to the machine and start it, and it's really easy to maintain. So we, in the following presentation now, we want to show you with two examples how we used the dragonfly discovery for assay development for our high throughput screening, and I will go now a little bit into detail where I explain to you how we did set up a kinase assay with the dragonfly. And Adrian later on will talk about protein-protein interaction assay. So the first example is a kinase. So this kinase is phosphorylating a certain substrate under the consumption of ATP. We made here use of the HTRF expertise from SysBio. So we made the phosphorylated peptide visible by a simple thread assay. And in this assay, we were interested what is actually the activity of the protein that we had in our hand, and what is the Michaelis-Menten constant of ATP. So for this, what you have to do is basically you have to titer the kinase and you have to titer the ATP in your assay. I want to show you now how we did this with the dragonfly discovery. So a very nice thing here is that we already could do this experiment on a 1536 well plate, because we didn't need to dispense here anything by hand. And what we have done, we titered on this 1536 well plates from left to right the kinase, and from top to bottom we dispensed the ATP. We did this for our DMSO nutrial control, and then we made a duplicate and did this with an inhibitor control, which shows us our lower assay window. As what you can see here, that we didn't use the whole plate, so the parameters that we wanted to check, we didn't need the whole plate. So the rest of the plate, which is not colored here, we could use then for a next experiment. So the kinase we did in 10 concentration steps and dispensed it from 2 to 30 nanomolar, and the volumes that have been dispensed here were 200 nanoliters up to 3 microliters. The ATP concentration was 2 to 30 micromolar, and the volumes again 200 nanoliters to 3 microliters. So what you can see here, how the dragonfly actually works and how you titer two different reagents at the same time. And this is how the raw data then actually look like. So on the bottom, you see in green our full compound control, and in the upper part, you see the DMSO control, so the nutrial control. And what you can also see is that the values are changing from left to right, so from high values to low values, and also from top to bottom. How this is basically goes in line how we titered the ATP and also the kinase. So what we have done here, we did actually one experiment instead of 3 experiments. So we looked at the enzyme activity by titering the enzyme. We looked at the Km of ATP by titrating the ATP. And we already did the miniaturization step because we started in 1536 well plates. I want to show you now some actual data here. In this graph, you see the plot for 3 different ATP concentration. I have mentioned before that we did a 10-step ATP concentration titration. I used here for this graph only 3 concentration to make it easier for you guys to watch. What you immediately can see, the higher the ATP, the higher the assay window. So in red with the 30 micromolar ATP, you have a bigger assay window than using in violet purple 2 micromolar ATP. When you look at the green graph, it's 10 micromolar ATP. You can see that actually up to 10 nanomolar, you are kind of in a linear range in your reaction. And at 10 nanomolar kinase, you are actually having a pretty good assay window of 4. So now I have plotted the 10 micromolar, the 10 nanomolar kinase. I plotted this with the ATP titration, and you can see a nice Michaelis-Menten kinetic, and the Km value that we have here is 9 micromolar. So what we have determined here, we can use this assay using 10 nanomolar kinase, and if we want to work at Km concentration, we use 9 micromolar of ATP in our assay. A nice advantage of the dragonfly discovery is that you can not only use it for assay development, you can actually also use it for compound screening in a higher throughput. So since we have this assay already developed here on 1536 well format, we used this assay now also for routine compound testing. And what you can see on this slide here is a 1536 well plate, and on the we used here only the left side of the plate, and we tested compounds here. What we have here in the second row, and then on the very far right, we have in this column our DMSO nutrial control. Next to this, no, the animation is not working. We have the inhibitor control, and in between these controls, we have the dose responses of the compounds in duplicates. When we now look how the data actually look like, we use here one compound, and the dose response looks like this. And we can nicely see a pharmacological dependence here, and the IC50 is roughly 30 nanomolar. What you can also appreciate is that the assay noise is really low here, and the calculated Z prime is roughly 0.9, which is super good. And we have our assay window of 4, which is in line with our quality criteria. So I showed you here now how we use the dragonfly discovery in setting up a functional enzymatic assay. And I will hand over to you now to Adrian, and he will show you an example how we developed an assay for protein-protein interactions.
Yeah, thank you, Nina and Carrie, and welcome everybody from me as well. I will present you more of a practical view on the dragonfly, starting with the assay principle used in the upcoming slides. In our experiments, we tested a protein-protein interaction via TR-Fred signal detection. And this means if the proteins are disrupted by a small molecule, the signal drops. The goal is to develop a stable assay for HTS, as mentioned before. So in order to speed up our assay development, we first switched from 384 well plates to 1536 well plates early on. Now we had plenty of space to test various parameters. For example, the buffer specifics, enzyme lots, time course, different substrates, or DMSO tolerance. The next steps were combined matrix experiments like Km values of substrates or detection step optimization. For these experiments, the dragonfly was really useful as it had nearly no restrictions on what detergents to use or other buffer ingredients. Because of the switch to the 1536 well plates, I was no longer dependent on multi-channel pipettes and in fact only used pipettes to prepare the stock solutions, resulting in fewer single-use waste. However, we encountered a problem with some proteins binding to the syringes. As you can see on the left side, there was a slight difference using the dragonfly and doing the experiment by hand, resulting in a lower Fred signal. So we searched for a workaround and found one in a brief pre-incubation of the syringes with a BSA-enriched solution, and the assay worked just fine with the dragonfly again. In conclusion, we can say that using the dragonfly discovery, we saved a lot of time in our assay development cycle, starting with the usage of 1536 well plates from the beginning, coming to multi-parameter testing and using the DFD in various places and occasions, finding a way to combine assay development and screening. Currently, we are testing design of experiment approaches that hopefully show the same promising results as before. Yeah, thank you everybody for listening. And this was Nina and Adrian from Merck. We're coming now to the questions and answer section.
Yeah, thank you very much for that interesting webinar, Nina and Adrian. So now let's move on to the last part of today's webinar, which is the question and answer session. Okay, so the first question we've got is, what is the time saving in hours if traditional assay development is compared to assay development assisted by the dragonfly discovery?
So this of course depends on the assay, what you are going to set up. But as an example, I mean, you have the kinase assay that I described to you, where we basically did a multiple experiments in one. I would say using the dragonfly discovery, we saved 2 days. At least, yeah.
Brilliant, thank you. Okay, so on to the next question. We've got, would you recommend the dragonfly discovery also for screening applications?
This also depends on the assay. So if for instance, so it really depends. So if you have an assay which has a complicated kinetic, so an assay where the reaction is still going on while dispensing, I would not recommend the dragonfly discovery, because if you compare the time, how long it takes to dispense one plate, it's much longer compared to a multi-drop system. Yeah, and then if you would use the dragonfly, you would kind of see drifts on the plate. If you have an assay which is where the reaction is over, which is stopped, then it's fine to use it. Yeah.
That's great, thank you, Nina. So on to the next question. We've got, what is the advantage of working in 1536 well plates? When would you not use this format?
Mm-hmm. So, I mean, of course, the advantage of the 1536 well plate is simply the throughput. Yeah. When would I not use it? That's a good question. So I think I would not use it if I would have problems with the volumes. So sometimes you need, if you have some kind of detection reagents where you need higher volumes, that of course you cannot use it. I think cell-based assays are often tricky in this, especially if you are doing co-culture assays or are working with primary cells, then 1536 might be tricky as well. But the most simple advantage of the dragonfly discovery is the throughput. Yeah.
Okay, that's great, thank you.
And reagent saving, I have to say. So, of course, if you think about the wells are much smaller, you need lower volumes, then also you need lower amounts of reagents in the assay. That's another one.
Okay, brilliant. Okay, so the next question we've got is, do you also use DFD to make compound dose responses or only reagent distribution?
Okay, so if this is meant by this, so we use the dragonfly for reagent distribution, that's right. The compounds itself, we never dispense with the dragonfly discovery. For the compounds, we are still using our ecosystems here. For DMSO-based solutions, we are using the echo. However, for all kinds of other reagents, we are using the dragonfly. Yes.
Fantastic, thank you. So the next question we've got is, have you used the instrument to run cell-based assays?
Yes, indeed, we did. And since the dragonfly is working fast, you sometimes don't even have to be cautious of the cells that they settle in the syringes. So it worked just fine. Yeah.
Perfect. Okay, so the next question we've got is, are there dragonfly tips available that have low protein binding properties?
Yes, yes and no. So there are tips, actually only one generation of tips are now available that you should use. These are really low in protein binding. However, what we experienced is, Adrian has mentioned this before, that it's good to saturate the syringes with BSA, so you can get rid of any unspecific binding to the syringes as such. And what we also have done before, because we had some issues also with the old generation of tips, we have coated them with silicone spray, for instance, and then you got rid of anything else, yeah. But the syringes that are available right now are low protein binding, yeah.
Perfect, thank you, Nina. Okay, the next question we've got is, can assays done on the dragonfly be performed on liquid handlers as well?
Yes. Of course, there are different liquid handlers out there. We have a couple of different here also in our labs, also small benchtop devices, and each of these different benchtop devices, they have their pros and cons. Of course, we have also bigger systems like Beckman systems, and these assay can also be run on bigger machines. However, the programming of these assays are then, I guess, much more complicated. Yeah, but you can also use other liquid handlers. Yes.
That's great, thank you. Thanks very much, Nina and Adrian. So that's all we've got time for today. So thank you very much to our experts, Nina and Adrian, for that informative discussion and presentation. And thank you to everyone joining us online for your interesting questions. I hope you found this a worthwhile session. So please do be aware that you can download your certificate of attendance by clicking on the resources tab of the webinar. And if you've got any other questions, please feel free to email me at editor@selectscience.net and I will follow up with your questions. And if you would also like to listen again to the webinar or invite a friend to listen, it will be available to watch on demand in a few days. So goodbye and thank you once again for joining us.